Hepatitis B virus, commonly referred to as HBV, is a major global public health concern and remains one of the leading causes of chronic liver disease worldwide. HBV is a small DNA virus, but despite its size, it demonstrates remarkably high genetic variability. This variability arises from the virus's unique replication process, which involves a reverse transcription step that lacks effective proofreading. As a result, mutations occur frequently, leading to the emergence of genetically diverse viral strains over time. This genetic diversity has led to the classification of HBV into 10 distinct genotypes labeled A through J. These genotypes differ by more than 8% in their genetic sequence and are not randomly distributed across the world. Instead, each genotype shows distinct geographic clustering shaped by historical migration patterns, modes of transmission, and population dynamics. For example, genotype A is commonly found in Africa and parts of Europe, while genotypes B and C are predominant in East and Southeast Asia. Genotype D has a wider distribution, being present in Africa, the Mediterranean region, and parts of Asia. Other genotypes, such as E, F, G, and H, are more geographically restricted, occurring mainly in West Africa, the Americas, or specific regions of Asia. These regional patterns highlight the importance of understanding HBV genotypes within local and regional contexts. Beyond geography, HBV genotypes are clinically significant. Different genotypes have been shown to influence disease progression, viral replication levels, likelihood of developing liver cirrhosis or hepatocellular carcinoma, and response to antiviral therapy. As such, genotyping is increasingly recognized as an important component of comprehensive HBV management. Turning to the African context, Africa bears a disproportionately high burden of chronic hepatitis B infection. Millions of individuals across the continent are chronically infected, often acquiring the virus early in life through perinatal or early childhood transmission. Chronic infection frequently remains asymptomatic for many years, allowing liver disease to progress silently until advanced stages are reached. Despite this high disease burden, data on HBV genotype distribution in Africa remain limited, particularly in sub-Saharan Africa. Many countries rely on generalized global estimates rather than locally generated evidence. This lack of genotype-specific data poses challenges for clinical management, public health planning, and the optimization of treatment strategies tailored to local populations. In Kenya, and specifically in Western Kenya, available information on circulating HBV genotypes has historically been scarce. This gap in knowledge underscores the need for studies that systematically investigate which HBV genotypes are present, how common they are, and how they relate to important virological markers. Such information is essential for informing clinical care, guiding treatment decisions, and supporting national hepatitis elimination efforts. In summary, HBV is a genetically diverse DNA virus with 10 globally distributed genotypes. Africa carries a high burden of chronic infection, yet remains underrepresented in genotype-specific research. Understanding the local distribution of HBV genotypes, therefore, is critical, particularly in high-burden settings, to improve patient outcomes and strengthen public health responses.